These are AP Biology notes on Chapter 12, which is the cell cycle and cell cycle control. So remember that one of the characteristics of living things that all living things share is the ability to reproduce and to reproduce more of their own kind. And so that's what cell division is all about, basically, the continuity of life, or cell division. This beautiful picture shows the progression of the cell's nucleus as it passes from interphase through prophase and prometaphase to anaphase and telophase, and the formation of two new daughter nuclei. And so we'll look at each of these in turn as we talk about what happens. In unicellular organisms, then the cell division is actually the reproductive, the way they reproduce. Multicellular organisms have cell division for lots of different reasons, including development from a fertilized cell, like happens from the zygote or the fertilized egg cell as an multicellular organism grows, for growth, for repair. Whenever you have an injury of some kind or a tissue death, then cell division is necessary for repair. It's a very important part of the cell cycle, but the cell cycle is not all there is. It's not just about cell division. It's about the whole life of the cell. So here we see some pictures show examples. Of course, reproduction, when you have a single-celled organism, that's how they reproduce. Growth and development from one cell to multiple cells. And then, of course, for tissue renewal as you have cell injury and death. Most of the cell division that occurs in living things produces genetically identical daughter cells that are identical to each other and to the parent cell. The exception to this, of course, is meiosis, which is the cell division that produces sperm and egg cells. The cell's genome is the DNA of the cell or of the organism. The genome of a single-celled organism, like a prokaryote, might just be a single DNA molecule to lots of DNA molecules that you find in eukaryotic cells. In eukaryotic cells, the DNA molecules are definitely packaged into chromosomes, multiple chromosomes that have to undergo certain changes in order for cell division to occur in an organized fashion. This is a picture of a cell, animal cell, that is getting ready to begin, or is beginning the process of mitosis. This cell is in prophase. You can still see the nuclear membrane, but you definitely can see the chromosomal bodies within the cell. Eukaryotic chromosomes are made of chromatin, or they're called chromatin when you can't see the chromosomes. And this is a complex of DNA and protein. There are several kinds of proteins, including histones. And basically what happens is that each organism has its own characteristic number of chromosomes, but generally speaking, in most of the cell's life cycle, you don't see the chromosomes as chromosomal bodies. Somatic cells, or body cells, have two sets of chromosomes. That's called diploid. And gametes, or reproductive cells, have half as many chromosomes, which is called haploid. When it is time for the cell to reproduce by mitosis, the DNA must be replicated. During the normal everyday life of the cell, the DNA is kind of loosely wound and spread out more or less so that it's easily accessible for the purpose of transcribing the DNA into messenger RNA to produce the various proteins and other control molecules needed in the cell. However, when it's time for cell division to occur, it is necessary for the DNA to condense and form compact bodies so that you can easily check to make sure that each daughter cell gets a full set of chromosomes. When the DNA gets replicated, but in part of the cell cycle called the S phase, then each DNA molecule is basically duplicated. And then when the chromosomes condense, what you see is a chromosome with two sister chromatids, which separate during cell division. They're attached at a place called the centromere, and that's the place where they are most closely related to each other. Here we have a picture of a chromosome before mitosis occurs. The chromosome is composed of two sister chromatids attached or joined together at the centromere. They are identical to each other in terms of the DNA having been copied. As you remember from learning about the process of DNA replication, remember the two strands of the DNA molecule separate and each side is copied separately from the other, so that each strand or each molecule of DNA is composed of one old strand and one new strand, so that pretty much guarantees they're going to be identical to each other. During cell division, then those sister chromatids are going to separate and move into two separate nuclei. Once they separate at the centromere, then they're called chromosomes rather than chromatids. So here we have just an illustration of what we just mentioned, the chromosomes. In an individual chromosome, if it were condensed or at the beginning of the cell's life, consists of the chromosome arms, basically, with a narrow waist part called the centromere. When chromosomal duplication occurs, DNA replication and condensation produces the chromosome that is composed of two sister chromatids. So we have two DNA molecules, basically. And then when cell division occurs, those chromatids separate into two separate chromosomes into two separate cells, and so each one has the same amount of DNA that the parent cell originally had. Mitosis is the part of this cell cycle or the cell division where the genetic material is separated or divided, and cytokinesis is the division of the cytoplasm. Now, of course, there is a variation of cell division called meiosis that produces the gametes, which are non-identical daughter cells, having only one set of chromosomes, unlike the daughter cells of mitosis, which have both copies of the chromosomes. The cell cycle consists of that mitotic phase, the M phase, and a period of time called interphase. And during interphase is when everything else happens in the cell other than mitosis. So as the cell is growing, doing its everyday job that it needs to do, preparing for cell division, all of that part is called interphase. The interphase is about made up of three subphases, which together account for about 90% of the cell's life cycle. The G1 phase, which is the first gap phase, the S phase, which is synthesis or the synthesis or copying of the DNA, and the G2 phase, which is the second gap. Please realize that during the cell cycle, the cell is continuing to do its normal everyday job that it needs to do. It is growing, it is synthesizing molecules, it is doing whatever things that particular type of cell has to do. Even while it is preparing for cell division to occur, the regular everyday job of growth and maintenance of homeostasis are continuing. The chromosome duplication occurs during the S phase of the cell cycle. When we look at a graph to show you pretty much what happens in the whole cell cycle, remember that we're starting with one cell, it is growing, and then synthesizing the DNA, and then preparing for cell division, and then finally this small slice here is where mitosis and cytokinesis occur. So about 90% of the cell's life is spent in interphase. There are 5 phases in mitosis. There are prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis is a separate process that occurs or overlaps the latter stages of mitosis, often begins about the same time as telophase or during telophase. In some cells it begins a little earlier, in some cells it begins a little later, but it occurs right at the end to separate into two daughter cells. Here we have pictures and diagrams and photographs that show you basically what is happening in the cell during each one of these phases. We'll look at a little more detail. So in interphase, we have the cell in its normal state. We cannot see individual chromosomes. However, this says here it's the G2 phase of interphase, so that means the chromatin has already been duplicated. But you still can see the nuclear membrane, you can still see the nucleolus. You can see the centrosomes or the centrioles that have been duplicated here. In prophase, the chromosomes start to condense, and you can see that the chromosomes are consisting of two sister chromatids attached to the centromere. The spindle is starting to form. There's an aster or some short microtubules around the centriole pairs, which are starting to separate and migrate to opposite poles of the cell. In prometaphase, you can tell that the chromosomes are even more condensed than they were here in prophase. The nuclear envelope has disintegrated or fragmented, broken down, and we see that the spindle fibers are attaching to the centromeres of each chromosome. The ones that attach directly to the centromeres of the chromosomes are called kinetochore microtubules, and then there are other microtubules in the spindle that are called non-kinetochore microtubules, and they do not attach to the centrioles of the centromeres of the chromosomes. In metaphase, all of the chromosomes have been attached to kinetochore microtubules. The spindle is fully formed, and the chromosomes are lined up in the center of the cell along this imaginary line called the metaphase plate. There is at this time a tug of war basically going on between the kinetochore microtubules from one pole and the other that are attached to the centromeres. When the centromere splits under the force of that, as well as an enzyme that separates the two chromosomes from each other, we have anaphase. During anaphase, the kinetochore microtubules are pulling the daughter chromosomes from the metaphase plate toward the poles. The cell is also lengthening due to pushing of these aster microtubules against the cell membrane. And then in telophase, we see the formation of new nuclei. The chromosomes cluster at one end. They begin to lengthen and decondense. The nuclear membrane begins to reform. The nucleolus begins to reform. And usually about the same time, cytokinesis begins. In an animal cell, you see a cleavage furrow here that is caused by contractile proteins that will basically act like a drawstring, cutting the cell in half. These photographs just are the same thing that we just looked at on the in the pictures. In Interphase, you can't really see anything as far as the chromosomes. You can see the microtubules there in yellow. But in prophase, you can definitely see chromosome bodies inside the nucleus. In prometaphase, they're no longer inside the nuclear membrane, but they still look relatively disorganized as they're attaching to their spindle fibers and getting lined up. In metaphase, they're lined up in the middle of the cell, in the center of the cell, along the metaphase plate. In anaphase, they've separated the daughter chromosomes, the sister chromatids have separated and are being pulled apart. And then in telophase, we have the formation of two new nuclei and cytokinesis at the same time separating the cell. Now the spindle is made of microtubules, and that's really what controls the chromosome movement. Animal cells, plant cells, all kinds of cells have microtubules. All eukaryotes depend on microtubules of various kinds to separate the chromosomes during this process. In animal cells, the microtubules are organized by the centrosome or the centrioles. The centrosome replicates during interphase and forms two centrosomes, and they are going to migrate to the ends of the cell during prophase and prometaphase, and the spindle forms in between them. The spindle is composed of various microtubules. We have an aster, which is the short microtubules at each centrosome toward the outside of the cell, and then the spindle includes the centrosomes and the spindle microtubules as well as the asters. There are two different kinds of microtubules involved in the spindle. We have kinetochore microtubules that attach to the kinetochores on the centromeres, and then we have non-kinetochore microtubules that do not attach there. At metaphase, when the chromosomes are all lined up, that's when this tug of war basically is going on between the kinetochores from both poles. This diagram shows the kinetochore microtubules attached to the kinetochores. Usually there are more than one kinetochore microtubule attached to the kinetochore. The number depends on the species. It can be anywhere from 1 to 40 or so, depending on what the organism is. The non-kinetochore microtubules overlap each other, and basically there's some ratcheting action of motor protein action on them as well when the chromosomes are separating, when the cell is lengthening. So here we can see the kinetochores in a little bit closer view. It's a protein complex attached to the centromere where those microtubules attach, and basically that's what allows them to pull. In anaphase, the chromatids are going to separate and move along those kinetochore microtubules to the opposite ends. The way this happens is that those microtubules are actually shortening by a depolymerization process at the kinetochore end. Here, an experiment was done to figure out whether they were getting shorter at this end or in the middle, and so basically they tagged some of the spindle fibers with a radioactive or fluorescent tags so they could see what was happening, and what they found was that mark didn't move, and neither did this decrease, but what was decreasing was the distance between here and the centromere. And so that led them to believe that there was a depolymerization process going on at the centromere end. Basically, there's a protein down here, a motor protein, that moves this along, and then the depolymerization process that happens here within the kinetochore. So it kind of breaks down, it kind of pulls back and then breaks down at this end, as it's moving along the microtubule in this direction. Non-kinetochore microtubules are going to overlap and push against each other, and that's going to help elongate the cell. And then in telophase, you've got these identical daughter nuclei at opposite ends of the cell. The chromosomes begin to decondense, kind of loosen up from their tightly wound structures. And then cytokinesis begins during that period of time, and the spindle eventually disassembles in the process. In cytokinesis, we've got a couple of different things that happen. In animal cells, it occurs by a process called cleavage. There's a cleavage furrow, basically, that is contractile proteins that can pull against each other. In plant cells, a cell plate forms to separate the two daughter cells. Here we have the cleavage furrow in the animal cell. And we've got a contractile ring of microfilaments that are basically pulling like a drawstring to separate the cells from each other. In a plant cell, we have the formation of a cell plate, and this occurs as vesicles that are produced by the Golgi apparatus migrate to that metaphase plate there and begin to fuse together, and that forms the cell plate separating the two cells, making the cell wall in between the two cells, and then the membrane formation follows that. Okay, here we have pictures. These are onion cells dividing, and so this just gives us another view of what the cells look like as they're dividing. We see here in prophase, we can see the chromosomes beginning to condense. We can still see the nucleolus, and we can tell that they're all within the nucleus. In prometaphase, you can see a little bit more detail of the chromosomes. They're more tightly wound. Oftentimes you see them moving into position in the cell. In metaphase, you see that they're lined up here in the center. In anaphase, they're being pulled apart. And in telophase, we have a cell plate forming. We have two new daughter nuclei that are forming. These nuclei look very similar to this one right here, so it's kind of a reversal of the process. And these are just closer views of the same thing. We see the details of the chromosomes condensing, the nucleolus, etc. And the chromosomes here within the nucleus becoming more distinct, as they are finally very distinct here. Notice that you can tell that there are two sister chromatids attached together there. And then in anaphase, they're being pulled apart. In telophase, they're forming two new nuclei, and the cells are separating from each other. The way this is regulated is by a series of molecules in the cell. The frequency depends on the kind of cell it is, the frequency of division, and then there are various molecules within the cell that regulate when this happens. Cancer cells are ones that figure out a way to escape those controls in the cell cycle. There are specific chemical signals in the cytoplasm that you can see. The original experiments that were done to show this showed what happened when you fuse together cells at different phases of the cell cycle to form a single cell. So in one experiment, they fused two cells here, one in the S phase of the cell cycle and one in the G1 phase. And what they found was that the G1 nucleus immediately entered the S phase and the DNA was copied. In a second experiment, we've got one cell that's in the M phase already of mitosis, and we've got one in the G1 phase of interphase. And when they were fused, the spindle formed and the chromatin condensed into chromosomes, even though the chromosomes or the DNA had not been duplicated. So there's some kind of chemical signal that stimulates these processes to occur. There basically is a cell cycle control system, and there are several checkpoints in the cell cycle where the cell cycle will stop unless there's a go-ahead signal that's received. It looks sort of like this if you put it on the diagram. Okay, so we have our G1 checkpoint. The G1 checkpoint is like, okay, if everything's ready to go and the cell's a certain size and there's room to grow and we've got everything we need, then we can go ahead and start getting ready for cell division. That stimulates the cell to continue on through the S and G2 phase. There's another checkpoint at the end of G2. It's like, okay, is everything ready to go? And then if that's okay, then it goes into the M phase. And then finally the M checkpoint, which is at metaphase, generally says, okay, is everything ready? Can we go ahead and separate? The answer is yes. Then it goes on and continues to separate and form the daughter cells. For a lot of cells, the G1 checkpoint seems to be the most important. Most of the time when the G1 signal says go ahead, then everything else continues. If the cell doesn't receive that signal, it can enter a non-dividing state called the G0 phase, and this occurs in cells that don't need to actively increase in number or don't need to, or not growing a whole lot. There are some cells that stay in the G0 phase for years and years, like nerve cells. Don't need to really divide all that much. Muscle cells don't divide all that much unless they need to, unless a repair needs to be made somewhere along the way. So they'll stay in the G0 phase for quite a long time. So if the cell gets the go ahead, it goes on into the S phase, and if it doesn't, then it goes into the G0 phase. There are two kinds of proteins that are involved in this. They're cyclins and cyclin-dependent kinases. Remember, cyclin-dependent kinases, any kind of kinase, is going to work by causing some action to occur, oftentimes by phosphorylation. The CDKs, the activity fluctuates because it's controlled by the cyclins. And then when the cyclin concentration gets to a certain level, and forms a complex with the CDK, the cyclin-dependent kinase, that produces something called the maturation-promoting factor, and that's what triggers the cell's passage past the G2 chain point into the M phase. So here we have the increase in the cyclin concentration progressively through the S and G1 into the M phase, and then we have a drop-off in the production of cyclin. The MPF activity begins when the cyclin gets to a pretty high point and peaks at the same time as the cyclin does, and then falls off. And that shows the fluctuation of the concentration of those compounds. What's happening in the cell cycle is that the CDK, notice that it's got a pretty much constant concentration, but as the cyclin is produced and forms a complex with the CDK to form the maturation-promoting factor, that's what stimulates the passage into mitosis. Once mitosis has begun and pretty much on its way, then the cyclin starts to break down, releasing the CDK, and that's what stops the process from continuing. There are internal signals and external signals that can affect this. Internal signals would include things like the kinetochores not being attached, or something wrong with the DNA copying and so forth. Some external signals would be growth factors that either stimulate cells to divide or not to divide. There are some specific ones like platelet-derived growth factor or PDGF that stimulate the division of cells in culture. So if we put the cells, if we're trying to grow the cells in culture, basically there's not really, the free fibroblasts don't really grow very well unless you add the PDGF, and when you do, then it forms that substrate that it can grow on. External signals would also include things like density-dependent inhibition. So when the cells are crowded, they stop dividing. If you remove cells from the culture, then there would be a signal that would say, okay, we've got an empty space here that needs to fill up, and that would stimulate cell division along that area until the cells come in contact with each other again, and then it stops. There's also something called anchorage dependence. Animal cells mostly have to be attached to a substrate. They can't just grow like in a broth culture like bacterial cells can. Cancer cells are different because they can continue dividing whether they're inhibited by density or anchorage. They don't need either one of those things to continue dividing. The anchorage dependence shows the substrate they may have to join to. The density dependent means that they're going to stop dividing until there's a break or an open place. And cancer cells are going to keep on going whichever way. Animal cells, normal mammalian cells will grow in a single layer and then stop. Cancer cells will continue to build up. Cancer cells don't respond normally to these control mechanisms because of various things. Okay, some of them make their own growth factor, so they don't need to depend on a growth factor from surrounding cells or from the organism as a whole. They have something wrong with their cell cycle control system in one way or another. And there are several different things involved in this. There could be tumor suppressor genes that are not working properly. There could be proto-oncogenes that become activated and become oncogenes. Various things can make this occur. The process of converting a cancer cell to a cancer cell from a normal cell is called transformation. Your immune system oftentimes takes care of killing cells like this before they get a foothold, but sometimes they get missed along the way, and so they'll start to form a tumor. When the abnormal cells remain only at that original place, that's called a benign tumor. But when it invades into surrounding tissues, that's when it becomes malignant. And if it invades into the circulatory system or lymphatic system, that's when it can become metastatic. So here we have a tumor beginning in a gland, the glandular tissue here with the breast as it shows here. When it begins to invade neighboring tissue, that's when it would become cancerous. And then as it spreads into the lymphatic vessels or the blood vessels, that's when it becomes metastatic, forming tumors in other parts of the body.